This lesson will be looking at proof that homology is just grasping at straws, how it can't actually prove macroevolution and is only assumed to do so. But just before we get into anything, first of all, what is homology? For those who have no clue what the heck I'm saying, it is the idea within the idea of phylogeny that claims similarities in structures and functions between kinds prove that they have a connected common lineage. We will do for this lesson is we will take the top three examples of homology, which are vertebrate embryology, which is about similarities in embryonic developments between vertebrates, and then vertebrate forelimbs, specifically the skeleton, and then human and chimp genetic similarities. Now, with vertebrate embryology, you've probably seen these famous drawings, like in your school textbooks, that are used to say that, see, look at how similar these embryos are at the same stage. These kinds must be related then. Well, these drawings were fabricated by a man by the name of, if I'm pronouncing it right, Ernst Haeckel. See, below, here's some real images of these embryos, and below that, some far more accurate drawings of different kinds of vertebrate embryos at three different stages. And do you see how different these drawings really are from each other? Even at the earliest stages, still a pretty fair amount of difference. Pretty obvious. And with fewer cells, we would obviously expect there to be a more similar look. Obviously, the less cells there are, then similar creatures are going to look a lot more similar than they do with a lot more cells. That's just basic anatomy and logic. And those gill slits you're probably told about only exist in fish, though you were probably told that they existed in all vertebrate embryos. What they are is what are called pharyngeal slits. So in fish, they become gill slits, but in the other vertebrates, they become wholly different structures and functions. Now, with the vertebrate forelimbs, they're spoken of as if they're so similar, so basically the same, that these kinds must have a common ancestor. Well, are all the bones the same kind of bone? Yes, but they are drastically different in shape and proportion. Look at the purple bones. The humans is basically a rectangle. The horses is kind of like a triangle, and then is a long, skinny rectangle that then bends. The orcas is a small rectangle with this indent. The tortoise has a major bend at the beginning, then it goes into a long line that starts to curve right here. The frogs is sort of a rectangle, but with an odd bump here, a skinny point here, and a really weird end right here. Then we look at the birds. It has very trapezoid-like ends, and with a slight bend right here. Now when you look at the blue bones, okay, humans, very long, very skinny. Then you look at the horses, okay, you got this massive spur right here, and then you have this notch here, and then it gets really skinny right here. Alright, then we look at the orcas, it's a bunch of two tiny rectangles. Then we look at the tortoises, you got one that's really skinny with a trapezoid end here, and then this one is a lot broader with kind of trapezoid-like ends as well. The frog, you just got this one rectangle right here. Then the bird, you have a really skinny bend at the end here. and then wide, a lot more bent too. See, it's really open. You have this huge open space, unlike the others, except the frog maybe, excuse me, tortoise, but it's really compressed. And look at the orange bones. Okay, the humans, you got this kind of compressed rectangle right here. The horse, you got like a small square here. The orca, you got this weird hodgepodge right here. The tortoise, you got this one massive piece here. The frog, basically can't see anything, it's just right there. Then the bird, this one tiny little thing right there. And finally, looking at the yellow bones. Okay, humans, you got the fingers, you know. Then the horse, okay, you got what's basically a continuation of the blue piece, looks like that. Then you have this bend, and then you have the hoof. A lot different. The orca, I mean, look at the proportions here. Look at how long this is, and how it curves, and how very misshapen it looks like compared to human fingers. And look at how many more bones. Then you look at the tortoise. Okay, a little more similar to humans, but a lot longer bones, a lot sharper ends. Proportions are definitely different. The frog, less pieces, and they're a lot longer. Then you look at the bird. Okay, nothing looks like this. None of these other four look like that at all. So I mean, do these bones really seem that similar? I mean, they have very relatively similar structure and only very relatively similar function. Now, with human and chimp genetic similarities, genetic homology is not at all as simple and clean as it seems, and it's not evenly spread throughout the genome, which is the total of the genetic information. So let's look at how much difference there is between human and chimp genomes. First of all, there's around 4% total difference between the two, not 2% or less, which you're usually told. That equals to over 65 million base pairs. And if you don't remember what a base pair is from high school, it's those pairs of letters you see in the picture on the right. And what are these letters? Well, you think about each letter on one side as a letter in a book. The DNA being the entire book, and the genes that those letters make up being the words. Also, there's a 1.23% base pair substitution difference, which equals to around 35 million base pairs. And a substitution you see on the left of the new image, where you have, for example, a G at one place in one genome, but in the same place in the other genome, you have an A. That would be a substitution. And then there is a 40 million to 45 million base pair insertion/deletion substitution, which is an example on the right, where you have a letter in one genome at a certain place, like that G, but then in the same place on the other genome, you're missing something, or vice versa. That would be an insertion and deletion. And then at what are called the telomeres, which are the ends of the chromosome and a very unique role and function, there is around 13,000 base pair difference, which, because of how telomeres function, is even more significant than if we're in, say, the red area of the chromosome. And then when it comes to the genes, there's a 775 difference in genes. We have that number of genes that they don't have, and they have the same for us. There's an entire chromosome set difference. Humans have one set of chromosomes less than chimps. That is a lot of genetic information. that we do not have. And then with the Y chromosome, there's an over 30% difference. So that makes us guys and our male chimp counterparts a whole lot different because of how much information Y chromosomes contribute for us. How much a deal that information is. And I mean, the list can just go on and on and on and so on of how much difference there is between human and chimp genomes. Not really that similar. And with the genetics, there's even more differences than just with the DNA itself, like the letters itself. There's also major DNA methylation pattern differences between the brains. And DNA methylation, look it up on your own, won't get into the details, that has to do with coding for proteins, has to do with your DNA function, has to do with getting the information to make something. So hopefully by now you see that homology is only true if you assume that it's true. Because similarities between kinds don't have to mean that they have a common lineage. And actually, similarities between kinds are even more expected if there is a common designer than if there were a common lineage. So think about it. If a man makes multiple kinds of machines, you'll definitely see commonalities between them. And even more so with more alike functioning ones, because they come from the same mind. So the universal unifying origin of all kinds, all organismal kinds, is in the creator's mind. It's not in the first creation, the universal common ancestor. And also, think about this analogy. Two men invented calculus, Newton and Leibniz, each in a different nation, hundreds of miles away, having no idea about what the other was doing, or really even much at all about the other. So why can't you have two kinds have no connecting origin except within the creator's mind, no physical connecting origin, and yet have incredibly similar structures and function? So I'll end on this. Which should we believe in? Homology, which is a concept that isn't based on anything in scripture, but on pure assumption of truth and complete straw grasping, as proven, or phenomenology, which is a concept that's taught in multiple parts of scripture and concluded from not just faith in scripture's accuracy, but also rationalization and empirical study, instead of on total unsupported assumption.